Hi, good afternoon, everyone. I hope I'm audible. Uh, just now, I have shared a link in the chat box. So you can go to that link and start with the opening activity. Uh, Meanwhile, others can join in. Once you're done, you can share a screenshot in the WhatsApp group or directly message it to me. I hope you had a good afternoon. I hope you all ate your lunch. Let's get back from the activity in the next 2 minutes. I'll wait for 2 minutes till everyone is in the class and done with the activity. Okay, I have received Mahika's response. That was fast. Shrutika, Disha. Okay, Shrutika's I have received. Disha's also. Good, it's very quick. Quick recap of your unit 2, that is structures of biomolecules. Once you're back, just mention in the chat box that you're back so that I get a sense of how many of you are still in the activity. Okay, Mansi is back. Disha is back. Sandhya, I have shared a link with you in the chat box. You can save that link for later and solve it. That was just a starter activity so that I can wait for others to join in. No need to worry about not being able to solve it. It's okay. Just save the link so you can see what the activity is later. Okay, so most of us are back. Then that means it's time to share my screen. What is on my screen? On my screen is my slides. Okay. So, like I told you, in the last class, we didn't get time to discuss about kite graphs. Today, we will talk about kite graphs, how to draw the graph and how to interpret it. Now, usually, whether it is the new syllabus or the old syllabus, in Cambridge, they don't actually ask students to draw the kite graph, but they definitely give questions which have kite graphs which needs to be analyzed, and you have to describe and explain the graph, and different questions can be asked from the graph. So knowing how to draw the graph will actually give you a sense of what is the meaning of those kites in the graph, what information is it giving, and what variables you're taking into account while filling a kite graph. After that, we will discuss briefly about the main concepts of cell signaling and answer some questions about cell signaling. So our topic begins with kite diagrams today. Let's begin then. Okay, so today in the kite diagram discussion, I will be taking you through someone's case study, someone who has actually done a field work and is converting the data into a kite diagram and trying to get information from that kite diagram. And this is, I have also, I can share the link of that case study which I have used today after the class. So this is the alpine forest of New Zealand, and Mr. Darren used a belt transect to record species of plants found in interval of every 2 meters along this hill or mountain, whatever. So a belt transect, that image on the right hand side of your screen is for you to recall what a belt transect looks like. First of all, we use a transect for systematic sampling. So systematic sampling is mostly done when you want to study the relation between an abiotic factor with the species that you are studying. That species is mostly a plant species, or the vegetation of that place. How does it change with the environmental gradient? Okay, the environmental gradient could be from sunlight to shade, or from more salty region to a less salty region. Along that gradient, how is the vegetation changing? So Mr. Darren chose to study how the vegetation of these alpine forests, these are coniferous forests, how they change along the slope of a mountain. So the gradient was actually, I think, altitude, height. With height along the slope, how the vegetation is changing every 2 meters. So this is Mr. Darren. He is actually, can you see the yellow meter tape? So he's setting up this belt transect. Belt transect means two transects are parallelly taken along the same patch or same slope, and then quadrats are placed in between the region of these two parallel transects intermittently or periodically, keeping same gap of 2 meters. So actually, Mr. Darren did a belt transect as well as a point transect. So here in the picture, he's doing a point transect. That is, he has taken a 20-meter belt transect, and he stretched it, tied two ends of these two belt transect to two different trees from the starting to the ending point. And then whichever tree or shrub or plant is touching the transect in every 2 meters, he has counted them. Okay, so how is he taking the data? Every 2 meters he is stopping and checking which plant is touching that 2 meter point on the belt transect, and he has recorded them on a table. Mostly he has found these species. So if you remember, in systematic as well as random sampling, you have to have a key. So he used an encyclopedia or an authentic key book, identification key book, to recognize these plants, and he found that along that 20-meter transect he found this kind of grass called the tussock, then the bog pine kind of tree, then he found mountain daisies, these are shrubs, inaka are again shrubs, and mountain beech are the tall trees, okay, along the transect. So because these were the 5 different species of plants that he noticed, he, in his data table, he has 5 columns for these 5 species of plants. And if you remember, he was measuring the transect at an interval of 2 meters, and at every interval of 2 meters he was measuring, or he was counting how many species of these 5 plants are touching that point. So the main column, that is your independent variable column, is distance of every 2 meters. So plus 2 meters. Remember, in your data table you need to have the details in a very short and precise manner with units. So distance plus 2 meters means every 2 meters. He has placed the rows in the under the distance column as 0, 2 meters, 4, 6, 8 meters, 10 meters, 12 until 20 meters. And in every 2 meters, he has taken into account how many mountain daisies he found, how many tussocks he found, how many bog pine plants touched that 2 meter mark. Whichever 2 meter mark it touched, he counted and placed them in the box. And the same for mountain beech. Okay. At this point in this table, is there anything that is confusing, or you want to ask something from this table? Okay, then I will ask if you don't have questions. So my question is, what does it mean in the mountain beech column when there are so many zeros and then there is a 2, 3, 1? So what can you tell about the mountain beech distribution, species distribution along the transect, looking at the mountain beach column. What information can you give me looking at this table? You can type your responses in the chat box. Looking at the mountain beach column, what information can you give me about the distribution of this mountain beach along the transect? Waiting for your responses. Distribution is unequal and frequency is low. Thank you for giving a general statement. I'm looking for citing of data. Sandhya says it shows the mountain beach was only present between 14 to 18 meters, very precise on the transect, and it is not evenly distributed. Okay, that's a good description. Mostly found at higher altitudes. No, Shrutika, you cannot say that the 14 or 18 meter is higher altitude or lower, because I did not give you the information that whether he started the transect from low to high or high to low. The only parameter over here is distance. Okay, so along the slope, maybe the 14, 18 and 20 is downwards. Does that make sense? So that information is not known to us. So you can just say how many meters from the starting point had no beech trees, and from which meter to which meter you found those mountain beech trees. Yes, Mansi, they were found only after 14 meters. Okay. Okay, now I want you to just take 5 or 10 seconds to look at other species distribution. Look at mountain daisies distribution, tussock, bog pine, inaka. And tell me, from all these 5 species, which species showed more population? Or the frequency was higher? You can either say that population was the highest or the density was the most. Any thoughts? Mountain daisies, okay. More mountain daisies, inaka and mountain daisies. So over here, I would have looked at the higher value. So if I'm looking at inaka, the frequency is very high. That means I'm finding inaka at 4 meters I'm finding inaka up to 16 meters. But in case of mountain daisies, frequency is not as high as inaka, but the density after 8 meters is very high. And how did I know that? Because none other species were found more than 8 in number at one point. So at point 12 meters and 14 meters, daisies were the highest in number, 8. But no other species at any other 2 meter point had so much population. So I'm going to say that mountain daisies were more in number along the transect, even if they were not evenly spread. Okay, now Mr. Darren also has to keep in mind that his table has a title. Look at the title: Alpine plants found at each distance along the belt transect. Okay. Yes, Inaka's population is higher along the belt, but at one particular point, the highest population shown is by the mountain daisies. Okay, you are right. So Inaka as well as mountain daisies population is the highest, but the density at any one point, if you see, The highest among all 5 species is mountain daisies. No other species have more than 8 individuals at 1 2-meter point. Okay. So I was talking about never forgetting to give a title to your data table. Similarly, now Mr. Darren wants to convert this data into a kite diagram to understand more, to extract more information from this much data. Right now, he only has data about every 2 meters, which species was found in how many numbers. So how can he extract more information, more patterns and trends? So he definitely sets up the y and x axis on a graph paper. And if you are also doing a field work where you want to convert your information into kite diagram, you will need your pen, paper, graph, everything with you. And the next thing that they do is find the most common species. So this is how he describes most common species that we discussed over here. Is how do you decide which species is the most common? Not by how frequently it is appearing, but on an average in one point, large number of individuals, which species is showing highest population at any point along the 0 to 20 meter. And for that, we see that it's the mountain daisy. No other species has 8 individuals at any particular point. Even the second highest is mountain daisies, that is 7 individuals. Okay, so let's just extract and look at the mountain daisy part of our table. Okay, and we will begin our kite graph using that data. Okay, so let's see how we'll draw our table. So this is my graph. Sorry, I have taken my x-axis, and I have to make sure that along with drawing the x and y axis, I am taking equal intervals using a scale, not freehand. And then I label my x-axis as distance along the transect, and in the bracket I have to mention the unit. Have I taken distance as meters, centimeters, or kilometers? Then my y-axis are the species. Okay, now the tricky part is when you're drawing a kite, your kite will be narrow at some points, it will become broad, and it will become narrow again. So you have to estimate how much gap you should keep for each species. Now we saw that the highest number of species are the daisies in our previous slide. That means when I draw my graph, each of my species will have one central line, and there should be 4 boxes above the central line and 4 below. So overall 8 centimeters. So in my graph, if each grid is one, if each small square is say 0.1 or 0.2 millimeters, then one small box, or one, this box that I am showing with my cursor, this is 1 centimeter. Okay, will it be 1 1 2 3 4 5? Yeah, about 5 centimeters, not 1. This one box is 5, and then I take 4 centimeters above and below, and I just choose central lines for each. So each of my species, I'm going to take one central line, but I have to be mindful that above and below that central line, I have 4 centimeters difference. Okay, so that my kites don't overlap. Mountain daisy's kite should not touch Inaka's kite. Okay, so I'm going to take a width of 8 centimeter. Please remember why I took 8. Because I looked at my data and daisies were the highest number at any random point from these in my x-axis. 8 was the highest number. So I can actually take 8 or 10, anything more than 8. Okay. Next. What is my next step? My next step is I start plotting. Okay. Not very difficult. Let me tell you how I have plotted. So this is actually my graph. I have drawn the graph like this. So I saw that at position 0 meters, there were no daisies. So For mountain daisies, my plot line is this central line, okay? And how have I chosen the central line? I have made sure that from my x-axis to the central line, at least there is 4 centimeter distance. And above also have kept 4 centimeter distance before the next species starts. Okay. So at 0 meters, no daisies. So this is my first plot. At 2 meters, again, no daisies. Second plot is here. At 4 meters, 6 meters, no daisies. Then comes 8 meters. At 8 meters, I found 3 daisies. So how am I going to plot that? So my plotting will be 3 centimeters overall, 1.5 above the line, 1.5 below the line. So when I take 1.5 centimeters above and below the line, total I get 3 centimeter. Okay. So I got 2 plots. Now let's look at 10 meters. At 10 meters, I found 4 daisies. Okay. Look how I have plotted. At 10 meters, if I found 4 daisies, then I'll take 2 centimeters above the central line, 2 centimeters below the central line. These are my plots. At 12 and 14 centimeters, I get the highest number of daisies, that is 8. So I'll take 4 centimeters above the central line, 4 centimeters below. So make sure that the data that you have on the table, it's split into half above and below your central line. So that when you draw the whole line, that is equal to your data on the table. 14 is also 8. So I took 4 centimeter above the central line, below the central line. Always remember to have a central line for your species. Don't use the x-axis as a central line. Then your kite will go out of the graph. Okay. Look at 16. There were 7 daisies found in 16 meters. So I took 3.5 centimeters up, 3.5 below. 3.5 plus 3.5 is 7. That's what our number is on the table. Similarly, 18. At 18 meters, I found 5 daisies. So I'm going to split it into 2.5, 2.5. I'm going to take a point 2.5 centimeters above my central line and 2.5 below. Last at 20, I got 4 daisies. So 2 and 2. This is my last plot and this is my 2.5 plot. Now can you guess what should the next step be? Anyone? It's very simple. What are you going to do next? Shrutika? Correct. Connect all of these points. Now, this is where students make mistake. They think, okay, all these are 0, 0, 0. Let's start drawing from here. Wrong. You will, if you have made the central line with a pencil, you will start again drawing from here with the pencil and start plotting so that you get a boundary and it will come out as a nice kite shape. So overall, like how I have plotted for mountain daisy, I took 8 centimeter gap between each species so that every species get a 4 centimeter above and below. And I've made central lines for all the 5 species. So now my x-axis and y-axis are ready. Each species central line is ready. I have plotted each data of mountain daisy. Next, I'm going to join the plot, and while joining the plot, never use freehand scale. With the scale, you will start joining the plots. So mountain daisy plots are joined. After joining the plot, you will notice that at 20, your plots are not reaching the central line. That means at 20, you are still finding some species of mountain daisies. So your kite will end with a vertical line. It will end blunt. Do not manipulate your data and make it look like a designer kite. Whatever the data is, your kite should look like that. Then shade the inside of your kite. Okay. In exam, you will only be allowed to use pencil. Here I'm using different colors for different species for you to observe. But in exam, if you are asked to draw a kite, you will draw it in pencil. And no dark shading is allowed. You can only use pattern shading. So kite diagram for mountain daisy is done over here. Take a look at how the kite diagram for Inaka, Tussock, Mountain Beech and Bog Pine is drawn over here. All these plots were taken in a similar manner, looking at the table. So if for Tussock I have taken 2.5 boxes up and 2.5 boxes down, that means this was 5. There were 5 Tussocks at 6 meters or something like that. Okay. And then with distance, the number of Tussocks are decreasing. So at this point, you saw the data table, and you looking at the data table, you could tell whether the species was evenly distributed or not with a number. Now you have a picture. Now you have a diagrammatic representation of the species distribution. So this is more of a visual representation of the species richness and evenness. Okay. For our case, we can actually study the species diversity and analyze data with just one kite diagram. We can compare these 5 species distribution, and that's the purpose of a kite graph. Kite graph will tell you that along an environmental gradient, over here our environmental gradient is distance. Okay, distance along the transect. So you can actually place this line transect along a seashore. So this kite diagram will tell you that along the seashore, if you're finding 10 different species of aquatic plants, how are their populations varying? How are their own populations varying along the seashore? And you can compare their population with other aquatic plants population along the seashore. In this case, you can compare the population of bog pine and its distribution along the transect with its within itself, within the transect only. You can also compare bog pine's population with mountain beech population distribution, and with Tussock's distribution, and with Inaka and mountain daisy. So overall, looking at this kite diagram, you can tell that mountain daisies are more widespread than any other species, looking at the thickness of the or the height of the kite diagram. But looking at the length of the kite diagram, you can tell which species is more widespread or more frequent. You can see them more frequently. There's something in the chat box. Give me a moment. No. Yogeshwari has asked, is it okay when a point overlaps? For example, a point of Inaka and a point of mountain daisies are joined. No, Yogeshwari, that is a mistake from my end. I should have kept 1 or 2 more centimeters as a buffer, because I didn't do that. I had this overlap where my Inaka is touching the mountain daisies kite. That should not happen in an ideal graph. That's good point. Make sure that between your graph there is enough space, and to make sure that is happening, that's why I chose the highest number, that is 8 daisies, and I kept 8 centimeter gap. But while keeping 8 centimeter gap, somewhere I made a mistake. That's why my Inaka is touching the mountain daisy. That should not happen. Does that make sense? Okay. I will now move on to what do kite graphs tell us. So take a few seconds and read through these points. If you want, you can actually sketch notes on what kind of information is the question looking for when they are giving you a kite graph. What do they want you to extract information about? So kite diagrams allow researchers to graphically compare different frequencies of different observed data along the same transect. So my transect data will give a different kite diagram. Yours will give a different. I can compare both, and I can study how much similar our kite diagrams are. Within my kite diagram, I can compare the species distribution of these 5 species of alpine forest plants. Second thing I can do is, as the frequency is shown by an area in the graph, potential and hypothesized relationship between different observations can be more identified. This is very interesting because I want to take you to the previous slide and tell you how that happens. Suppose you are making kite diagrams of these alpine plants, and you can actually make a hypothesis by saying that the area where tussocs grow, mountain beeches don't grow over there. You can clearly see how the place where tussock spread is tapering or widening and then gradually narrowing. That is the point from where mountain beech population is actually increasing in your 20-meter transect. Do you notice that? Or you can hypothetically assume that maybe there is some sort of a relation between bog pine species distribution and tussock. Remember, these are still hypotheses. So mostly ecologists use kite diagram as the beginning of their research. They begin with this hypothesis using the kite diagram, and then they do further research to see. They go to the field and actually see, is there any competition between these two species, what are common about these two species. So it helps them understand more about this plant rather than just looking at their species distribution. This is a starting point to start thinking about more questions. Is there any relation between these species or not? Secondly, sometimes along with kite diagramming these plants, you can also make kite diagrams of animals found by placing putters or animal traps along the transect. You can find correlation of which kind of insect is found more around bog pine or around tussocks, and is there any relation between those animals and these plants. So this kite graph is a foundation to start thinking about these questions, hypothesizing some relationship and trying to prove them in your further research. That's what the second point here is. Third point, their most common, though not exclusive use is for showing the frequency. Just you look at the graph and questions around which species frequency is the highest at 5 meters, or between 5 to 10 meters, which species were completely absent. Just to understand that species frequency and take a survey every year, different ecologists use the kite diagram for that purpose. Okay. Do you want me to pause for a moment so that you can take note on this, or should I move ahead? Please let me know in the chat box. Okay, I'll wait for a minute so that you take notes on what is the significance or what kind of information do you get from kite graphs. Just let me know if you're done. Okay, great. At this point, do you feel that you understand kite graphs better, or whatever I shared was known to you? Can you just unmute and tell me if you learned something new or it was repetitive for you? Okay, Yogeshwari says that she understands kite graphs better now. I Yeah, clarified your concept about kite graph. Once we had done a kite graph question from the harder textbook. Okay, Ranu also says she has a better understanding. And I felt that just exposing you to a graph and then asking you to find the meaning of that graph, it's too many new things at the same time being asked. So understanding how to draw the graph will actually tell you what is the meaning, what is actually plotted on the graph. Once you know those variables, then you'll be able to get more information or study trends or patterns in that graph. Okay. Thank you for sharing, Mahika and Dania also. I will now move to the next slide where I want to tell you that Mr. Darren could also do another type of kite diagram where only you are not just plotting the frequency, you can also plot percentage cover of your 5 species of those alpine plants. So basically what I'm trying to say over here is when you're drawing a kite graph, the kite graph can tell you how many individuals of your species were there. So you're calculating species frequency, but you know that species diversity can be calculated in many ways. You can calculate species diversity by calculating the density, the frequency, and the percentage cover. So with density, you saw how the kite diagram can be drawn, but with percentage cover of your species also you can draw kite graph. Now remember that those species which are very difficult to count because they cluster together as colonies or their individual bodies are fused with each other in the colony, like mushrooms or lichens. Lichens are actually symbiotic relation between algae and fungi. So algae and fungi are symbiotically fused with each other, and they will form these uh, colorful or white or gray patches on moist rocks in the rocky seashores. So lichens are very difficult to count one by one. You can only count them as a colony. So previously also we discussed, we use the percentage cover type of calculation when you can't count each individual because either they are too many in number or they are too small and they get hidden under bigger species. So here is a table which is actually showing percentage cover of alpine plants, mosses, lichens, and small shrubs. They are very difficult to notice. So unknowingly, you get biased and start counting bigger plants, and you skip these small ones. So how to do a kite diagram of these? Again, you take your independent variable column, that is 2 meter distance along your belt transect from 0 to 20, and then you place your quadrat and count colonies, and with the number of small boxes in your quadrat gets covered by a moss, you count that out of total number of boxes in your quadrat. If your total quadrat has 100 boxes and 50 small boxes are covered by the moss, then you say that the moss's percentage cover at 0 meter was 50%. At 2 meters it was 100%. That means the whole quadrat was covering the moss colonies. At 4 meters again it became 50, and so on. Same goes for lichen. Nothing was found in 0 meter and 2 meter point, but from 4 meters to 16 meters lots of lichen colonies were found in the quadrat area. So these are all in percentage, remember. Small shrubs also have been, the data has been plugged in. Now, with this data, how will you draw a kite diagram? Same, you take this is just the data for say mosses. Okay, I'm not showing the full graph. Your x-axis is distance along the transect in meter, and y-axis is data only for moss. Okay, so you write moss over here and then you draw your central line. Remember, your central line represents 0%, and the space above and below will represent your 50% and 50%. So if in the first data I got 50 mosses in 0 meter position, that is from starting point, then I will only plot 50%. So will I plot 50% on top, or will I just plot one number, one X mark at the bottom, or should I take 25 above and 25 below? What should I do? Tell me in the chat box. I want to plot and show that at the 0 meter mark, I found 50 colonies of lichens. Correct. 25 up and 25 below. If 50% is shown by 5 centimeter, so 25 will be shown by 2.5 centimeters, right? So I'll take 2.5 mark here and 2.5 mark here. So my first plots will be on 0, note 0 meters. Correct. So this is how my first mark will be. Then second was 100. Look, I took 50% above my center line, 55 centimeter below, and then so on. So my kite, after plotting everything, this is how my kite diagram for moss, lichen and shrubs will look. But this time, this is not showing me species frequency. This is showing me percentage cover at every 2 meters. Take a few minutes, seconds to just look at the graph and see which species percentage cover is highest at. So you can't say overall percentage cover of one species is more because it's varying with distance. So can you tell me at 16 meters from the starting point, which species population is the, or percentage cover is the lowest? Lowest percentage cover of which species at 16 meters? You can answer in the chat box. Moss. Okay, everyone is saying moss, 16 meters. So for this question, first I'm going to find where is my 16 meter mark on x-axis. And for small shrubs, I'm seeing that from the central line, I will need a scale over here. Remember, if I'm asked by how much value, then I have to use a scale and measure this length. Say this length is 50 or 100%. So that means this will be 10 centimeter. This will be around less than 2 centimeter. And moss is the smallest because it's covering the smallest number of boxes. That is, where is, oh, it's 0. There's nothing at 16 meter here. So you can also calculate values using your scale, looking at a kite diagram. So kite diagram actually gives you better information instead of just having a numerical table. When you convert that number into diagrams, analyzing and comparing different species percentage cover becomes more informative. Here is another example of a kite graph of two species A and B is percentage cover. So this is just another way. I'm just showing you another way in which you can draw the kite graph of. So let's say a data is given to you and they have given you a blank space to draw the graph. So you can take 10 centimeters for species A, 10 for B, 10 for C, 10 for D. Make sure whenever you take any distance, it should be equal for each species and it should be big enough so that the kites don't overlap. And then you start filling the data. So at 0 meters, A's percentage cover was 25%. So then you always have each species own central line, which is the 0%, and 50% above and 50% below that central line. So if at 0 meters A was 25%, then you will take 2.5 centimeters above and below this 0 mark. That's taken here. Then next at 10 meters, 50 is the percentage cover. So again, 2.5 above and 2.5 below. Okay, not 2.5, this will be 5. So 50, yes. This is actually not 2.5, this is lesser. This will be 1 point something, 0.4. 0.4 above and 0.4 below. Okay, and that way, never forget to close your kite. Your kite may not end as a point, but end bluntly, but close it. Otherwise, your kite diagram will go wrong. Next slide, I'm going to show you. And you have to answer this question, okay? This is a kite diagram along a line transect of 25 meters. At what distances were most grasses and dandelions seen? Take a few seconds to think. Okay. Most grasses and dandelions. Yogeshwari, will it be the same for both? Watch carefully. What you wrote is for dandelion. Correct. Mahika, you are right. Ranu is also right. Mansi is right. Sandhya, right. Shrutika, correct. So basically, yes, Disha. Disha, for grasses, it is actually 20, not 17.5. And for dandelions, it is 5. Okay. So basically, you have to find answers from the graph this way. This is the simplest question. So most grasses were seen at 20 and most dandelions at 5. Okay, this is a bad kite diagram. Okay, I'm showing you a non-example of how to draw kite diagram. Now y'all spot all the mistakes and tell me in the chat box. What are the wrong things done in this kite diagram? Let's see if you can notice. Open ends, correct, Danya? So in the barnacles kite diagram, I can see there are open ends. Also in periwinkle, the ends are open. For periwinkle, there is no center line, correct, Shrutika and Sandhya? Any other? There are 3 mistakes. Very good, Yogeshwari. The graph is not originating, or the kites are not originating from the x-axis. Again, Disha, you're right. X-axis is not labeled properly. Like, what about the quadrat? Is it saying the number of quadrats or distance of the quadrat from the origin? Not labeled. Okay. Thank you for your responses. You have been responding very well. So I want to show you the answer to this. Don't know if it will be visible. Yes. So these are the mistakes. And the most important thing is title. The title is very vague. The graph has been named as Rocky Shore biotic factors. Which biotic factor? No one knows. And it's not short and it is not detailed, as in precise. Title does not contain much detail, no mention of where site is or date of the samples taken. Then some of the kites are open, like you all told. Does not give distances from the starting point or units. That is, x-axis is not properly labeled. And because there is no central line, the kites are not starting from the y-axis point. So you have to make sure that your kite diagram doesn't look like this. So after this class, I will share with you a very well guided practice question around kite diagram. You can actually go through it and solve it. If you want me to see, you can mail me your work. There is no deadline for this. This is for your own preparation. So I don't want you to feel that there is added work that I'm giving. This is just an additional practice, so that whatever you learned about kite diagram, you can actually execute that in that worksheet. You can do it whenever you have time, or whenever you're revising about unit 18, and send it back to me for checking. So this is all about kite diagrams. The next thing in our agenda was discussing about cell signals. We have about 20 minutes for that. So I just wanted to remind you that cell signaling is from unit 4 of your AS level, and I had added this in your revision of module 57 and 58. So after this class gets over, there are two things that you have to remember. Whenever we do any live classes, and I'm referring to some modules, it means that you don't have, if you haven't done that module, you don't have to do all of the tasks of that module in detail anymore, because I have already discussed that module in the live class. But you have to complete the assignment of that module, because that assignment is going to check your understanding. Okay, that is one thing. That is, live classes covering modules means you don't have to go back to Edmodo and do that module all over again. Second, what you have to do then, you have to only do the assignment part of that module. If that module doesn't have assignment, don't do it anymore. It's over. It is considered that it is done for you. Okay, and I will not go back and look for your submissions anymore. But if you have not submitted assignments, please do the assignments. That means if we are discussing cell signaling now, I'm expecting that at least by next week, or towards the beginning of next week, or by end of this week, you should complete the assignments in module 57 and module 58. Okay. All right. Let's check our understanding about cell signaling. In this animation, you're seeing this is the cell's lipid bilayer, and this red color structure is actually a receptor, and this receptor is nothing but a transmembrane protein. That means this protein spans across the lipid bilayer. On its outer face, it has a cup-shaped structure. This is a protein, and that cup-shaped structure is the receptor part, which is going to receive the signal, that is the ligand. Once the ligand binds with the receptor, there's a 3D shape change of the protein. And I want you to look at what is happening in the cytoplasmic side. This is the cytoplasmic side. Notice what is happening when the ligand is binding. The receptor is forming a dimer, means two different receptors are bonding together. On the cytoplasmic side, they are converting ATP to ADP and inorganic phosphate. That means they're breaking one phosphate bond of ATP on the cytoplasmic side. So on the cytoplasmic side, there are lots of ATPs bonded or hydrophobically attached with these proteins. They are getting broken down. When phosphate bonds break, energy is released, and that energy will activate this enzyme phospholipase in the cytoplasm. This phospholipase gets phosphorylated. Okay, that means they get that extra phosphate from the ATP. Look, it's getting phosphorylated. And this phosphorylated enzyme is activating another enzyme protein, and that another enzyme protein is again activating a series of reactions. So basically, what happens in cell signaling? There are three things that happen. First, the ligand will bind to the receptor, and this part is called the reception or receiving of the signal. After the receptor binds to the ligand, it will pass this new information that it has got while binding to the ligand to the other side of the membrane. Okay, and that is called transduction. And this transduction can only happen if the receptor is complementary to the ligand. Otherwise, the receptor and ligand will not form a complex and no message will be passed. Then finally, because a message is passed to the other side of the receptor, that will lead to a series of chain of reaction. Sometimes the chain of reaction involves activation of a messenger inside your cell. That messenger is most of the time cyclic AMP or CAMP, and it's called the second messenger. So if this is the second messenger, who is the first messenger? Your first messenger is no one but the ligand, or the chemical signal which is coming to the receptor. That is bringing the first message. So your ligand is the first messenger, and after receptor and the ligand form a complex, the other messenger which gets activated or receives a P from the ATP, or a phosphate gets added and it gets phosphorylated, that other messenger goes and starts activating other enzymes in the cell so that a new reaction will start inside your cell. So that other messenger is called the second messenger. Now, when the second messenger gets activated, then a series of or a cascade of reactions happen inside your cell, leading to a cell response. What could a cell response be? Most of the time, a cell response means either a gene gets switched on, means it will start getting expressed, a protein is synthesized, or something is broken down in your cell to simpler form. So either an anabolic reaction will happen or a catabolic reaction will happen because of the signal that came in. That whole pathway is called reception, transduction of signal, and response. So cascade of reaction, cellular response happens first. So when I look at a cell signaling diagram, three things are the outline of cell signaling. One, ligand will bind to the receptor, that is reception. Two, triggering, there is a trigger. Because of this receptor and ligand binding, a trigger is formed. Signaling will happen inside the cell. This is passing of the message or transduction or transmission of the message. Third, a specific cellular response will happen. So reception, transduction, and finally a cellular response. Take a moment to look at the whole annotated diagram. Notice what is happening at 1, what is the meaning of 2, and what is the meaning of stage 3. Okay, so we saw that signal transduction pathway will lead to a cellular response. Right now I'm just telling you different information points about cell signaling. In the next slide, we are going to look at an animation.
In our body, they work together in a coordinated manner and initiate different kind of tasks. So here we say this is communication between cells, which directs the cells what to do and what not to do. Second thing is that it regulates and modulates cellular functions in our body.
So here the person is actually narrating why cell signal is important, the significance, the need. Are you being able to hear it, or should I share the link with you?
Can you turn on the captions?
Okay. Because he's actually writing whatever he's saying. So if I turn on the captions, they will overlap. Let's see.
Third important point here to note is that it installs a signaling pathway between target cells and signal producing cells. And we see this process It occurs in both prokaryotes and eukaryotes. Now we will see what are the basic principle that lies behind cellular communication. We see here it's based on three core communication processes. First one is called reception, where the surface membrane or intracellular receptors receive signal from extracellular molecules. Second thing is here is that transduction. Signal transduction is called, where a modified signal gets transduced towards its target point. Mainly it's the nucleus of the cell. And ultimately we have cellular response, which may be activation, deactivation, or modification of certain cellular processes. The pathway of this communication process is as reception, then transduction, and then response. And it all occurs in a target cell, but it's initiated by a signal producing cell. When we see a cell, that signal receiving cell, it has got intracellular receptors as well as surface receptors. But in that case, we will see the cell surface receptor, or we can say membrane bound receptor.
I will pause the video over here to just check what he's talking about: target cell and a signal producing cell. So we saw in the beginning of the video that a cell always gets itself involved in signaling, so that it is not isolated. It is always communicated with its neighboring cells. So a cell signaling function is actually a part of your cell membrane's job. Your cell membrane has these receptors all embedded in its fluid mosaic structure, and your cell membrane takes the responsibility that the cell is not isolated from the neighboring. It's getting all the information from the neighboring cells, and the cell itself can send signals to others. So when a cell is sending signals or receiving signals, the whole process of signaling involves two cells. One cell is the signal producer and the other cell is the target. Means that cell will show the response. How? Because that cell will receive the signal. That's what he is telling. Then second thing that he told is the receptors can be of two types. Sometimes your receptors are embedded in your cell membrane, and sometimes your receptors are in the nuclear envelope. So if this receptor is on your cell membrane, you call it extracellular receptor. If your receptor is on the nucleus membrane or nuclear envelope, then it is an intracellular receptor. That means your ligand can actually enter the cell or get modified into another molecule and then reach the nucleus and then trigger a signal. Okay, two types of cell signaling can happen. One signaling where the signaling starts from ligand coming from outside and binding to receptor on the cell membrane. Another type of signaling is when your ligand is say a lipid, and we know that lipids can easily pass through a cell membrane because cell membrane is also made up of phospholipid. So if it is a small lipid molecule which is a ligand, it will pass through the cell membrane and its receptor is actually on your nuclear envelope. So that will lead to an intracellular signaling. Okay, right now he's showing an example of a cell signaling pathway which happens with membrane proteins on your cell membrane. So the receptors are on the cell membrane, they're extracellular receptors, or you can say they are transmembrane receptors. Sorry.
Communication. You know, we have different kind of cells present in our body. They work
Okay.
together in a coordinate.
Where were we?
Cellular. Or we can say membrane bound receptor. So a signal producing cell sends a signal via a molecule which has specific receptors for its reception in a cell. So here we see a cell modifies its receptor protein on receiving a signal through different mechanism, like we see we have the addition of phosphates or we have removal of it. It's a vice versa. And this in turn modifies attached signaling protein to it, and we get relay molecules which transduce this signal towards target site. And these relay molecules signaling constitute signal transduction, because these relay molecules are the molecules that gets remodified in a way to promote this signal towards a target site. and ultimately we get the response in the form of activation, deactivation, or modification of certain genes or cellular processes. So from reception through signal transduction and ultimately the response from the cell. This all constitutes the basic principle of cellular communication. So guys, this was all about principle of signal transduction in a really simple and basic manner. Guys, thank.
Okay, so maybe this is the place where you find it very vague. What is he actually telling? What he's telling is signals from outside the cell to inside are actually transferred either in the form of phosphorylation, means once the signal binds with the receptor, on the other side or other face of the receptor a phosphorylation happens, means an ATP will be broken down to ADP and inorganic phosphate, and that inorganic phosphate is a highly energized molecule or or a group. Whichever macromolecule it goes and binds to inside your cytoplasm, it will energize it. So that most of the time what happens, on this end there is an ATP which gets broken down to ADP and inorganic phosphate, and that inorganic phosphate goes and activates an enzyme which was initially resting or inactive. Now that the enzyme got active, it will search for substrates and it will start catalyzing a reaction which was not getting catalyzed earlier. And because that reaction is getting catalyzed, new products will be formed. Those products can go and stimulate other reactions to happen. So this is the cascade of reaction. And this phase of cell signaling is called signal transduction, where activation of one molecule leads to a relay or amplification of activation of other molecules inside your cytoplasm. And because that leads to a relay of message, gradually, ultimately, it will lead to stimulating the DNA to either express a gene or to break down a protein or to release some kind of enzyme or secretion. That is always a cell response. Okay. So before we start discussing about the multiple choice questions after this slide, there are about 3 or 4. Do you have any questions or anything that you remembered about cell signaling that you want to ask? No questions? Okay. Let me tell you that in the next session, I will be doing some past paper questions, worked examples, which are already there in your module 58. So you can actually wait and attend that session so that your module 58 will be done in the live class itself. But right now, I'm just going to ask you basic questions about cell signaling. So let's start that. We have a few more minutes left for the class and we'll be able to do that. So we have question number 1 and 2 on screen. Please don't share any answers now. I will cold call. So you read the question and be prepared with the answer for question number 1 and 2. Question number 1 is which cell receives the signal during cellular communication? The signaling molecule, the transducer, the receptor protein, the target cell, and the response cell. Second question is, place the following steps of signal transduction pathway in order from top, that is from the beginning, to the end, that is bottom. So you will number them in an order so that they will give you the correct sequence in which signal transduction happens. If you need to do some rough work in your notebook, take a few seconds. I'll wait for 30 seconds so that you're ready with your answers. Okay, everyone is ready with the answer? All right. So I will ask Yogeshwari, can you tell me the answer to the first question? So which cell receives the signal during cellular communication? Receiving the signal is always the target cell. Correct answer. You have shared the correct answer, Yogeshwari. So the answer is number 4, option number 4, that is the target cell. The second question is to sequence these statements into the correct order of signal transduction, and this will be answered by Ranu. Ranu, tell me what should the first statement be?
First statement would be a ligand binds to the cell surface receptor.
Correct. Next.
The next would be the receptor activates a protein at the membrane.
Yes.
Third is second messenger molecules are released, and
Correct.
the
Final
target
response.
causes.
Correct answer. So this is the answer to the correct sequence. All right, next question. Again, I'll give you 30 seconds. Specifically, what does the signaling molecule bind to in order to start the transduction? And the next question is, which is the final step in cell signaling? And both the answers will be given by Shrutika. What is the answer to number 3? What does the signaling molecule bind to in the start of a transduction? Shrutika, can you hear me? Yogeshwari, your answer is correct. No, Shrutika, it's not the transducer protein. Transducer proteins are those relay molecules inside the cell. To start the signal transduction, first the signaling molecule has to bind to the receptor or the target cell's membrane protein. That is, the answer is the receptor protein, number 3. Okay, not the signaling protein. It's saying what does the signaling molecule bind to. So the signaling molecule is the ligand, it will bind to the receptor. So answer is 3, option number 3. Okay, Shrutika, what is the answer to question number 4? Final step in cell signaling. Can you tell? Final step of cell signaling is cell response. That is option number 2. Correct. Yes, it is cell response. Okay, I think we have one more. Yes. So these are your answers. Last question. A cell releases chemical signals into its surrounding space in order to communicate with neighboring cells. Which of the following must occur for the communication to be successful? A chemical signal must bind to hormones on the neighboring cells. The same chemical signal must be produced within the neighboring cells. The neighboring cells must be able to absorb the chemicals through diffusion. The neighboring cells must have the right receptor to receive the signal. 5 seconds to think. And you can share the answers in the chat box. Which option is the correct answer? A, B, C, or D? Okay, Disha and Sandhya, Mansi, they all say D. All right, the correct answer is D. My question is, why is A wrong? Can you tell why A is wrong? Can you tell in the chat box, all of you? Why do you think option A is wrong? What is wrong with that option? I'm waiting for your responses. Okay, Disha is saying that it is not the signal but the ligand which should bind. Actually, Disha, the signaling molecule is called the ligand. Both are the same thing. So here when they are saying that the chemical signal must bind to hormone, they are meaning that the ligand will bind to the hormone on the neighboring cell. Sandhya, you are correct. Sandhya says hormones are not receptors. Correct answer. The mistake in understanding over here is that most of the time we forget to realize that hormones themselves are signals. Okay, so hormones themselves are ligands. They are received by the receptors. So when your endocrine gland is producing a hormone, it is poured into the blood and the hormones go to target cell, remember. So your hormone is not the receptor, your hormone is a signal. So why will a chemical signal go and bind to another signal? That should not happen. It should bind to the receptor. Okay, so all hormones are signals, they are not receptors. That's why A is wrong. B is wrong because if one cell is producing a signal and neighboring cells also produce a signal, what's the point? Signal has to reach the receptor, then only the cell will understand what is the signal about. If everyone is producing signal but nobody has the protein to receive the signal, no communication happens. Neighboring cells must be able to absorb the chemical by diffusion. If they absorb it, it will not produce any changes or relay of message will not happen. After absorbing, the lysosome may digest the chemical and the message will be lost. So C is also wrong. Okay. With that, our discussion today comes to an end. Thank you everyone for participating actively. I